It's really a pleasure for me to welcome everyone to our first ever virtual meeting. As Carolyn mentioned, we have a very good attendance registration this year. We have a great group of speakers, and although it's always nice to meet in person, I think that you're going to get a lot out of this meeting, including updates on all of our projects, our public health partners, our interns, and so I'm really looking forward to doing things a little bit differently this year. So what I want to do just for a few minutes this morning before we start the formal program is talk a little bit about some of the highlights of the year so far and some things to come in the next few months. So despite all the disruptions of COVID, and many of our projects remain not fully working at capacity due to COVID restrictions, we have accomplished a lot even since February and March when the restrictions started to take effect. One of the major goals we've had in the last year is to increase our internship program and also to reach out to surrounding states to improve our reach, the reach of our programs, and the inclusion of partners in the public health and academic sectors nearby. And we've accomplished both of those things together by recruiting new interns from several universities in the surrounding areas, including Taylor Ludwig, who you can see on the left there, flagging for ticks in Arkansas, Macy Lively from Southern Nazarene University doing mosquito trapping there in the center, and then Linda Wolf from Oklahoma University. And you'll be hearing a little bit more about their projects later in the meeting. A new project that we're gearing up to start very soon is what I think is a very exciting one that we're developing in concert with Saravanan Thangamani. Many of you probably remember that Saravanan was part of our center until about 2 years ago when he moved from UTMB to SUNY Upstate Medical University to lead a new program there in vector-borne diseases. And Saravanan has developed up in Syracuse a very nice program to take ticks submitted by citizens in New York State that are then sent to his lab for testing for a variety of pathogens, such as Borrelia burgdorferi for Lyme disease, Ehrlichia chaffiensis, and others, including viruses like deer tick virus. So these samples are tested by PCR, and then the results are fed into a data analysis system through a program called REBCap that some of you may be familiar with. And the location information is entered into a GIS database and mapping software. And eventually what comes out in the end is shown down here on the right and enlarged a little bit on this slide, is maps of the state that show the prevalence of different tick species, and most importantly, the prevalence of pathogens in those ticks. So you can see on the right here, for example, that different counties within New York State have different risks for these tick-borne pathogens. And not surprisingly, Lyme disease is the highest risk, with about 27% of ticks submitted being infected by Borrelia burgdorferi. The submission of the ticks is done through a very simplified system where packets are mailed to residents, including envelopes for submission of the ticks through the mail to the testing center and initiation of the whole process. So we're hoping that by early next year, we're going to have this system up and running, at least for parts of Texas, and ultimately we hope to expand this to surrounding states as well, to really get a much better handle on specifically what regions are at highest risk for tick-borne diseases in our region. So this is going to be a very nice complement to our current surveillance program for tick-borne diseases, and you'll hear a little bit more about it from Donald Boyer later in the program. The outreach and education programs have been diminished due to COVID, but they got off to a great start last fall. You can see the numbers of our 3-day courses conducted, our face-to-face classes, partnerships with various public health agencies to host these, and then pesticide applicator classes also conducted this year. So in total, even despite the COVID reductions in these programs, more than 1,000 people have been reached by these training programs, representing over 200 municipalities and public health departments. And Sonia Swiger will be telling you more about this program later in the meeting. But obviously, our training and outreach programs remain a real strength of our center. The new program that we started last year, a research program to look for tick and flea-borne infections along the Texas-Mexico border, is getting off to a little bit of a rocky start thanks to COVID, but a lot of progress has been made. So ticks are being collected and submitted for testing here at UTMB, especially the University of Texas, El Paso, led by Doug Watts, has been very active at collecting ticks and also fleas and submitting those for testing. And also UTRGV is participating in this program by preparing to start screening human sera that are part of a diagnostics program that they've set up on their campus. This also involves collaborations with Texas State Health Services, a variety of other animal control and animal shelters, veterinary hospitals, parks, residential homes, et cetera, down in the Valley region. And so just to show you a few of the data, you'll see more later, already in this program, we've detected several pathogens, including Ehrlichia and Rickettsia in ticks collected in the Valley. I also want to introduce to those of you who are not from Texas A&M, a new member of our partnership. This is Philip Kaufman, who's the new head of the entomology department at Texas A&M in College Station. Phil is trained as a livestock veterinarian entomologist. He began his career on the faculty at Cornell University and then moved to the University of Florida in 2005, where he became a professor with a combination of research, extension, and teaching activities there. His specialty has been in integrated pest management of livestock systems, beef, dairy, poultry, equids, and swine. Recently began working more with companion animals and wildlife. He specializes in biting and non-biting flies, including mosquitoes, as well as ticks. He was very active in the Southeastern Regional Center of Excellence program in Florida. He joined Texas A&M in July of this year. And he's now the principal investigator of the Texas A&M AgriLife portion of our center activities. So please take the opportunity to welcome Phil to our center and to Texas, and I'm sure he's going to make a great contribution to our program through his leadership up in College Station. Finally, you're going to hear a little bit of information about the future of the program. We learned just very recently a little bit from Jeff Borchert about plans for a new funding opportunity for the second round of the 5-year cycle for the centers. Probably that will occur in late 2021 or early 2022, with funding targeted for mid-2022. And we've reserved a few minutes at the end of the program tomorrow for Jeff Borchert, our program officer from CDC, to give us a little bit more information by looking into his crystal ball for the future of the program. So with that, we're going to move now into the regular program, and Carolyn is going to introduce our next speaker.
All right, thank you, Dr. Weaver. We'll now begin presentations with our keynote speaker, Dr. Rebecca Christoffesron. She's an assistant professor at Louisiana State University. If you could please share your screen, Dr. Christoffesron.
Great. Thank y'all so much for having me virtually here. I'm excited to talk to everybody. I know a lot of the people in the room. So today I'm going to talk about something that I've been thinking about for a while now, and that's disease X and how that fits into how we as vector-borne disease researchers really kind of approach things.
Dr. Christoffesron,
yes.
I believe you're sharing a different screen than what you intend to.
Oh dear.
So if you'll click the share screen button, you should be able to select the screen that you want to show.
I see. Is it better now?
I think we're seeing your email. That's why I asked.
Oh, well, that's not interesting at all.
Actually, it is very much so.
Oh, was it? Great. Is that better? Is that the same one? Oh, that's not me. That's Caroline.
Yeah, I think, Scott, you have to stop sharing your screen.
Oh, okay.
Sorry about that.
Yeah, I'm sorry about that. I believe it's at the top, Scott. There should be a small red box that says stop sharing.
I see chat, home, meetings, contacts.
Are you at the top of your screen?
I may have lost the big screen. I'm looking at the little one that has the time displayed near the middle.
Okay. Let me see.
It has a share screen icon, but doesn't seem to be responding.
I'll stop it from here. There we go.
Thank you.
No problem. And now we're ready, Rebecca, for you, Rebecca.
Try again.
Perfect.
There we go.
There we go.
Thank you.
All right, now you can see my screen. I'm still happy to be here. I'll talk to you about something that's been kicking around my brain for a while, which is the idea of disease, the next big thing, and how we should frame it with things that we already know from vector-borne disease. So in 2018, as I'm sure a lot of you know, the WHO put out for the first time this disease X on its R&D blueprint, which was really just sort of knowledge that there was something lurking somewhere that we didn't know was going to pop out and cause, a global pandemic. And in 2020, that seems very obvious, but in 2018, it was actually quite a interesting and daring move on their part. And so when we look back, though, at data from years past, we see that this was probably motivated by a novel coronavirus, or SARS-1, emerging in 2012, and then MERS coming out in 2013. So we had these new coronaviruses, as they happen, emerge, and we knew that these things were the next big thing was out there. And so disease X became kind of a focus of a lot of people's imagination and focus, because what is the next big thing? When the next big thing happens, when SARS-1, when MERS, and now when COVID-19 comes out, we always go into the basic characterization. We quickly respond as a community to determine, the basics of transmission, pathogenesis, therapeutics, and vaccines. But what about next time? How is understanding the disease X of today going to really help us prepare for the next time? For example, things that we still don't know about what's going on now is what led to the spillover? What's the reservoir? Had there been failed spillovers previously that we could have caught? We're still trying to kick around is it, a bat virus, or is it pangolins? Like, these are all the kinds of questions that we still haven't answered. And so we still don't know what happened this time. So how are we going to figure out what happened next time? And so that kind of got me thinking, or I was thinking about this before, but it really sort of codified this idea of disease of X, of F of X. And so if you haven't been homeschooling a middle schooler for the last 6 months, I'm going to remind you that F of X is a way to notate a function, which is just a relationship or expression involving one or more variables. This is the function that I've been fighting with my daughter about for the last 6 months, so I'm going to use it. But it's also the equation of a line. It's the basis of linear regression, and we all might just recognize it. So F of X is it's the multitude of factors that kind of go into what's going on. And my whole premise is that disease F of X then represents the understanding that without studying the ecological, the environmental factors that surround transmission, the multitude of anthropogenic factors that define the pathogen human contact, because that's really the drivers that we're looking at, epidemic or pandemic potential of a pathogen can't be defined, whether we have identified that pathogen or if we haven't identified that pathogen. So and we as vector-borne disease people kind of already know this, so I'm preaching to the choir a bit, but 75% of emerging infectious diseases are predicted to be zoonotic or vector-borne, right? And we already know this because we study it, but we know that there is the ecology that goes into the sylvatic cycle. There's potentially its own set of vectors, its own set of reservoirs, its own set of environmental drivers. We have the circumstances that are specific to the intermediate zone or the zone of emergence, that interface between animal and human cultures. And then we have the urban cycle, right? Or the anthropogenic cycle that really is again, a third set of environmental parameters. And so we have to understand all of these things, both independently and together, to really know what's the next big thing or what's going to happen with pathogens we've already identified. So for example, we have experience, right? So in 2014 and 2015, Chikungunya and Zika emerged as really large outbreaks in South America. And we had already identified, we knew probably more about Chik than Zika, I would argue at the time, but still we were kind of caught unawares in these new ecologies. So these things moved from its home base in the Eastern Hemisphere and found a new home in the Western Hemisphere and set up shop. And I'd argue that we haven't fully understood how or if these have set up shop in their own set of ecologies in the Western Hemisphere, or really defined what that would be. And then if we're looking forward to the next big thing, if we look at this data, right, which was very unsystematically put together by me, but we look at the past decade, vector-borne diseases like Rift Valley fever, West Nile virus, yellow fever, and then we throw in dengue and all the, the regular outbreaks, they represent a really large public health threat or ongoing issue. I mean, we've had a banger year for dengue, for example. So these are the types of pathogens, and these are the types of questions we have to get at if we expect to get in front of the next big thing, which I argue could very well be a vector-borne disease. Because meanwhile, in 2020, right, we have the first autochthonous dengue outbreak in Italy. We've got uptick in Usutu virus in the United Kingdom. We have the first West Nile infection was in the Netherlands. That was just days ago. Let's see. We have tick-borne encephalitis outbreaks in cheese in France. So we have all of these things that are occurring, and we have all these opportunities. But what we don't know is, when do these opportunities become public health crises? So what are the variables driving emergence? How do we get to this disease F of X? Well, we know that there's globalization, climate change, and human behavior. And these are just categories that I've put together. They're by no means, the end all. And we can sort of make a function, right, out of these things. So we can make a linear function. This is mathematically incorrect, just so the math people don't get mad at me, because all of these things are intertwined. And so none of these variables are really independent. There's all kinds of collinearity. But if we think about it in a theoretical context, we can put together this function of these things that we really need to know in order to get to solve for the answer, or to solve as best of the answer we can get, which is, how do we get to know what the risk is for the next big thing? So if we look at changes due to increased globalization, that's going to have its own set of circumstances to make a subfunction of land use change or agricultural needs, increasing populations. If we think about climate change, of course, we have increased emissions, we have temperature changes, we have deforestation, all of the things that are leading to just changes in the landscape. And then finally, human behavior, which I didn't probably appreciate as much until the pandemic, is really a driver of how these things are shaped up. We have political unrest, we have preconceived notions of researchers or of clinicians, and then of course we have to deal with the rise of anti-science, anti-GMO that might stymie certain vector control programs or efforts. So again, we have all of these little sub-functions that go into the big function. So I'm going to talk about some examples of these. This is an example of how globalization might change. This is from a paper in 2017. And you can see that land use is different. So if you have oil palm, I gotta move my little thing over here. Oil palm monoculture, that means it's an oil palm plantation like, or a polyculture, which means it's a multi-use kind of land use, and then rainforest. There's definitely a difference among where we find Aedes mosquitoes in Cote d'Ivoire. So we can see here, going back, we can see here how land use is really driving where we find mosquitoes, and that's going to drive where mosquitoes are in contact with people, and that's going to drive where pathogen comes into contact with people. Again, this is a brand new paper that I just love, showing in Europe how the distribution of Aedes albopictus was really correlated with major national roadways. And so you can see the frequency is not only is it more south than north, which makes sense, but it's really along this major highway as opposed to these smaller kind of roadways. So again, this is a human construct that is driving where we find these mosquitoes. And then of course, something that none of us are surprised at, megacities are making dengue deadlier. That's a bit of a sensational headline, but basically we're making more opportunities for transmission, in that we are bringing humans into contact with mosquitoes more, because we're creating more opportunity for breeding, more places for that contact to really happen and be in close proximity to where people live. If we talk about temperature, this is from Sadie Ryan at University of Florida. It came out last year, and this is showing that global changes in temperature are going to make more places suitable for Aedes mosquitoes for longer. And so as you increase the length of time that a certain place is suitable for mosquitoes, you're also going to increase the length of time it's suitable for transmission. So temperature is of course driving where we're going to see expansion of the vector, and that's going to lead to expansion of the pathogen, and is probably contributing to the first autochthonous transmission of dengue in Italy. Out of my lab, this is something we've been working on, just looking at shifts in behavior and how temperature causes mosquitoes to do things differently. The mosquito, each row is a mosquito and each column is a day, so it's 1 through 25, and you can see at 24, 28, and 32, the biting habits of mosquitoes, these are Aedes aegypti, are just crazy different, not only at the individual level, but at the population level. So each colored square is where they bit, and each white square is where they didn't, and we test it every day. So here you can see that the biting habits are very different, which of course then translates into differential transmission opportunities. And so when we put this into a simulation model, we get not only we get differences in total number of secondary human cases, so the amount of transmission to other people from mosquitoes, but also the percent of bites that resulted in transmission was higher when you have more overall bites than less, which is not surprising. It's somewhat intuitive, right? So, and then we have human behavior. So this is the seminal paper from Tom Scott and Steve Stoddard, looking at how people move the virus. So this is the thing where we figured out that people really were the moving force of dengue in neighborhoods and across places to move the virus, which was again a result of Aedes aegypti just being very comfortable in houses and not really wanting to work much. So this is an example of how human behavior is driving transmission. And I still think that idea has probably not trickled down into mass education programs for places where these Aedes-borne viruses are endemic. And then preconceived notions about what you test for, right? This goes into identifying where pathogens are and what's responsible for disease. So this picture comes from Rwanda, and it speaks to her project, my PhD student, Fausta. We went to Rwanda during the 2018 Rift Valley fever outbreak, and we found that a handful of cases that were otherwise clinically diagnosed as Rift Valley fever were actually not Rift Valley fever. They were from orthobunyaviruses. And in this case, they were from Bunyamwera. And we actually found that of the clinically diagnosed Rift Valley fever-like illness, only about 30% were molecularly confirmed as Rift Valley fever. And that tends to be the case. There's a lot of clinical diagnosis. I think that's why we missed the first couple of cases of Chikungunya and Zika, is because you've got dengue, right? Because everything kind of looks the same. Unfortunately, they all have these sort of generalized mild febrile illnesses in most cases. So when you look you find what you look for, so you diagnose what you find. So there has to be some sort of movement towards, when we find that it's not dengue, when we find that it's not malaria, then do we have a panel of, okay, what else are we going to go look for? And that's got to be prioritized by the function of where is the ecology of certain pathogens. I mean, we've all got those ArboCat books that have just hundreds, if not thousands of pages of identified viruses that we still haven't really characterized all of the ecology or the biology of it. So this all goes towards the vector-borne disease community, which is, we are uniquely positioned at the forefront of pandemic responses and really, hopefully, prospective pandemic responses, because 3 of the last major epidemics have been vector-borne. We've got Chick, Zika, I'd argue yellow fever, and then we're always working on endemic transmission, often big outbreaks of malaria or dengue or Rift Valley fever. And if you think about the basis of what we measure transmission of, which is the basic reproductive number, which everybody's an expert on Twitter now, but that has its roots in malaria, in vector-borne disease. And so vector-borne disease community really is, I think, positioned to take this idea of disease FFX and sort of mold it into how we go about preparing for the next big thing. Because we already recognize that disease X would emerge as a condition in multiple factors. This is from a paper that was recently published, but it was from a meeting a year ago or so, I think 2019. Time doesn't exist anymore, right? But we already recognize that this is just, there's so many things that go into describing how a virus is going to emerge as an outbreak or as an epidemic or as a pandemic, whether it be vector-borne or not. And so the thesis basically is that it's all a giant tangled mess, right? What is disease FFX? It's a giant tangled mess, but we are good at messes. We tackle these messes all the time with the viruses that we study because we have to. We have to speak to the ecology of the vector. We have to speak to all these things that I've talked about, plus the myriad of other things that I didn't talk about that goes into the function of how transmission and spillovers occur. And I think that was shown by how the vector-borne disease community responded to the current pandemic. I mean, we had people who belonged to the CDC Center of Excellence who turned around and started testing for coronavirus. We've all heard about Saliva Direct. Nathan Grubaugh at Yale put out, which helped the NBA stay in business and has been, I think, used in a couple of surveillance settings. Greg Ebel at the University at Colorado State. They did some really cool work about nursing home workers. My lab transitioned to a clinical diagnostic lab in early March of last year because we actually had a really big spike back in early March. We tested a little over 4,000 people in about 10 weeks, I think, and it was 18 medical facilities, first responders. And then, of course, we have people like Dr. Weaver, Dr. Plant, who helped kind of speak to this kind of mess of aerosols and whether or not coronavirus was an aerosol. So gave some legs to the much needed mask mandates and mask wearing. So the vector-borne disease community responded to this pandemic. And I think some people were confused, at least in my area, because they're like, we usually work on mosquitoes, which they don't understand they actually work on viruses, but I work on mosquitoes. But this was no surprise to anyone in the vector-borne disease community because we already knew what to do. We already knew how to respond to something big like this. So how do you solve this function, right? These are my opinions. I think funding agencies need to prioritize the science or surveillance. It tends to be not very sexy science, and I think it gets kind of a bad rap. It turns out to be very descriptive sometimes. I think the Centers for Infectious Disease are a very important first move. The Centers of Excellence for the CDC. We just have to make sure that these discrete centers, which are very important, do not lead to like a discretized thinking, because I think there's a lot of people out there that if we make a flow of information across a lot of different regions, then of course we just get diversity in thinking, which is never a bad thing. And I think engaging international scientists in their home regions to spearhead region-specific investigations. This is another thing that I think vector-borne disease people do very well. Just continue to do that, continue to really have strong partnerships, because if we know how a virus is acting in its home country, or its home region, whatever, that'll help us sort of know what to prioritize to look for should it jump like Zika and Chick into something new. We need to develop innovative methods for surveillance. Salivary protein biomarkers is very interesting, and people are using that. I think there's more progress to be done on that. Integration of data streams, like can we use biodiversity measures to really kind of look at, is there a point at which invasive mosquitoes like albopictus take advantage because we've cut down too many types of plants, or we've displaced too many other types of insects. We need to continue to break down discipline silos. Can vector-borne disease researchers, for example, inform urban planning or redevelopment? I know that there was some article recently about green spaces in Singapore and how that sort of affected dengue transmission, I think. And then finally, making data publicly available, but also using publicly available data. So Dr. Weaver just talked about what's going on at SUNY. That's wonderful. I think citizen science is becoming more and more useful with apps and verifiable pictures that you can just not take somebody's word for it that they got bit by a Aedes albopictus. iNaturalist is one that we've used at my house just for fun. And then there's a new app in Europe for mosquito watchers. I can't remember what it's named for it, but these public databases will increase collaboration and more creative approaches to really get to solving this function and solving the idea that we have to know all of the things that go into disease FFX, not just what's its name. So that's my spiel, which again, I'm probably preaching to the choir, but I'm happy to do it. And so with that, I will be excited to take any questions that you may have.
Thank you, Dr. Christoffesron, for that very topical and insightful presentation. Like you said, we'll now take questions for her. Dr. Roundy, did we have any questions from the audience or any hands raised?
Yeah, looks like Dr. Weaver has our first question.
Good morning, Rebecca. Thanks for that very nice presentation. You caught my curiosity with your experiments looking at biting frequency of Aedes aegypti at different temperatures in the lab. Can you tell us a little bit more about how you did those experiments?
Yes, I'm hoping, yeah, it's very exciting. So what we did was we took, we have Hematech, and we put individual mosquitoes in some little containers with screen, and we would thread the Hematech through to our environmental chambers so that they would never be removed because it's freezing cold in this building. So bench top is never the temperature you want it to be. And so we would look for blood meals. So this is a conservative estimate on biting. We have other data looking at a little bit more nuance into biting, but for this particular data, it was blood meal. So we'd look for the presence of bright blood, either partial or full blood meal in the belly, erring on the side of it's not real. So we trained people to look for it. We had the same person doing it every day, so we get rid of that bias. But yeah, we just look and see if they had taken a blood meal. And then the little dots, I don't know if you saw the little dots on the I think I messed this up. Hold on. Yeah, on the right. That was we checked for eggs as kind of a validation. And none of the mosquitoes that we recorded as having not blood-fed laid eggs. So that was kind of a check for us. But yeah, it was a really cool results.
Did you find the same kind of frequency of partial blood meals that are reported in nature?
No, I think I found more because I could monitor them, every mosquito every day. And these are lab mosquitoes. That's the other thing. So they're a little bit more, they have a little more predilection to biting. So I don't know that I would feel comfortable comparing them just because it is an artificial system and we know the same mosquito. We know it's the same mosquito over and over again. But I do feel like there was 4 or 5 mosquitoes in the 28 group that really bit a lot. Everybody else did like 3 or 4, which I thought to be pretty standard. So we're still kind of looking into that. The individual heterogeneity was surprising to me.
Thanks.
And interesting.
Any other questions, Dr. Roundy?
I actually had one kind of dealing with what the solution to this function is. are you imagining something like anticipating a specific agent spilling over, or something more comparable to the doomsday clock where certain events trigger us closer to a spillover event?
Yeah, I think it's the latter. I think I don't know that we can, I think it just depends where you are and when, right? So if we talk about Amazon deforestation, then, maybe it'll be an Amazonian virus. If we're talking about just mega cities, maybe it's something in Asia. So I think it's probably more the doomsday clock where just a set of circumstances is gonna, align and we're gonna have a spillover. My solution, I think, is more about really just understanding that we have to keep drilling down on the pathogens that we're looking at, as well as the ones that we kind of haven't been looking at. There's a lot of neglected arboviruses, bunyaviruses are fairly neglected, for example. And we can't just stop at we can't just stop at, we know what's going on in the urban cycle, so we're going to throw a bunch of vector control at it. I think we keep, that's totally what you should do right now, right? But I think you just have to get under it and get at the root, which is just understanding the entire ecology of these viruses vector pairings. So it's just, and what is the endpoint? How much is necessary and sufficient data to really understand and where you can stop? I don't think we know that yet. I think that's part of finding the solution, is figuring out what is the sufficient amount of data or types of data that we need to know to say, okay, we've got this one done, let's move on to the next one.
Yeah, it seems like this would take a lot of data.
Yep.
Looks like we have another question from Dr. Matt Dasha.
Hi there, Rebecca. That was a great presentation. I'm a clinician here at UTMB. I'm sort of like a wannabe vector-borne disease person, so I like hanging out with all you guys. So this is a great presentation. I run a One Health course with some of the other collaborators here as part of the center's network. And my question, you piqued my interest with your study in Rwanda, which I'm hoping is going to be published soon, because we're part of a consortium looking at undifferentiated febrile illnesses. And I'm wondering what your thoughts are on the use of some of these point of care sequencing technologies like the mini ion sequencer to be able to start characterizing some of these either potentially novel pathogens.
Right, so that is published in AJTMH. It came out in July of this year. So it was in cattle, I think, and some goats. I am all for point of care sequencing. I have a background in statistics, so I love data. So the more data, the merrier, in my opinion. I know that there are technical challenges to that. The mini-ion makes it easier, of course, because it's portable and it requires a little less of the logistics involved. But I am 100% on board with I would love to do that, because I think knowing where, for example, we even had some interesting results from that study where we got, when we sequenced, we got all three segments of Bunyamwera. So we were able to say definitively, like we got Bunyamwera, but then we would get two segments of Batai, or we'd get once we actually had some where we had Rift Valley fever confirmed animals that had sequenceable amplicon for one of the bunyas. So there is definitely room for us to go and think about what else is out there. And I think that would be a wonderful, start at this for sure.
Thank you.
We have our next question from Dr. Gabe Hamer.
Hey, Rebecca, Gabe here. Great job. So I've kind of, observed similar, things like you, looking at these emergencies, disease effects. One thing that tends to happen is a lot of federal and global health agencies dump a lot of money, basically in reaction to these emergencies, millions, billions of dollars. So what is our pitch to sponsors, federal sponsors of research, in terms of trying to be more proactive to maybe start doing research, before these things emerge? I mean, that's a challenging pitch, I think, and I've not perfected it. So I'm just wondering what your thoughts are on that.
Yeah, so I think it was early or mid-spring where I was having a conversation, and it might have been with you, VTech, where I was talking about like, everybody raise your hand if you've had a pathogen surveillance grant get told it's not significant and triaged. And I think the count was up to like 15 just within a few people. So it's not just the funding agencies, it's the scientific community who are reviewing grants, right? So I'm not sure. I mean, if this doesn't do it, right, if 2020 doesn't do it, I don't know what will. So I don't know if we, you know, I don't know if the vector-borne disease community writes a, you know, I don't know how much those op-eds work, but if I knew that, I would, that's the $1 million question. I would very much like to know, but I think the outbreak should give us some traction to make the point. Whether or not there are resources there to do it, I mean, I think one of the things is like, look, if we know it's out there, we can at least get to the diagnostic point, because the diagnostic delay for COVID was significant, even though it didn't seem like it was that long. It was a couple weeks, you know, I can't remember. And then just, you know, it was long enough. So perhaps you start with, you know, it would make your response faster, maybe.